as we leave cells, looking and focusing just on single cells, now we're going to begin to diversify. And we're going to talk about our first organ system and first organ. And this is one of those favorite biology trivia questions. What is the largest organ in the body? Because in many cases, when you think of organ, and when someone says largest organ in the body, you're thinking internal organs. But in fact, much like the plasma membrane of your cells is an organelle, your skin that covers your body is your body's largest organ, with in fact 16% of your total weight is comprised of your integument. Now, integument is certainly going to include your skin, but in addition to the skin, we're going to include various glandular components, various nervous system components. We're going to see circulatory components, your blood vessels coursing through the different layers of your skin. But we're also going to include hair and nails. Now, we don't just include those because they're on the surface of your body. We include hair, nails, and skin because they're composed of the same stuff. They're really produced by practically the same cells. And what these cells are filled with on the surface of your skin, the plates that together form your nails, and the cells that look almost like scales that form your hair, are all cells that are filled with an intermediate filament called keratin. In fact, when you look at the horn of a rhinoceros, that's keratin. When you look at the antlers of many animals that have antlers, that's keratin. And so it's amazing how we can have a surface that is, for most individuals, very soft and supple and pliable on the surface of our skin. And then we can have materials that are so hard as our nails or horns of animals made out of the same stuff. It's really pretty impressive. And so as we look at all of these different structures, all of these different components of integument, of the skin, of how they're put together, I'm going to refer back to what you've already seen in histology, right? Y'all went through histology pretty quickly, but y'all covered integument, the different layers. And so we're not going to spend a lot of time looking at it as much as we are sort of defining some of the parameters of it and explaining the significance of the integument and the layers and the cells that are present. So we're going to start with skin. And as we look at what the skin does, the skin not only provides an outer covering, the skin is not only something to look at, but the skin is also a means of protection. And we can see protection against injury, because I think you would wager that it's probably safer to cut or injure your skin than it would be to cut a blood vessel or a nerve beneath the skin. So in that sense, the skin and the fatty connective tissue underneath, it's going to be protection. Also against bumps, hits, bruises. But this second one may not necessarily be so intuitive to think of the skin as a component of the immune system. The world around us, especially the microscopic microbial world around us, there's a lot of quote-unquote germs that are looking ways to gain access to your body to make you a host for their reproductive life cycles. Our body is designed to prevent pathological infection, certainly bacteria being one of those. And what better way to prevent a pathogen from gaining access to the internal parts of your body than to provide an intact, contiguous physical barrier to infection? And that's your skin. Because many of these pathogens need to infect a cell, viruses for instance, need to infect a cell so that that living cell can replicate and that virus can multiply. Is that going to happen on the surface of your skin? No, because what are the cells on the surface of your skin? They're dead. So these pathogens have to dig their way through these dead layers of cells to try to get to the deeper layers where you actually have cells with nuclei that are alive. So you're presenting a physical barrier, but it's really an inhospitable environment for these pathogens. So your skin is part of your innate immune system. Now, as we've looked at cells and we've looked at DNA and RNA and protein and organelles, we still go back to that first unit and I ask you, what does your body mostly contain? Water. You are mostly water. And so your skin is going to be a waterproof barrier. When I say waterproof barrier, certainly when you go swimming, you don't swell up. Okay, water's not going to get in. But your skin, more importantly, prevents water from escaping. Your skin prevents you from drying out, or desiccating is a big fancy word for drying out. Prevents you from dehydrating. Now, if you're running around outside, it's 105 degrees, you're going to dehydrate. Because we have some secretions and some glands that are going to try to cool your body off, and it's going to be mostly liquid, and that could be a problem if you lose too much fluid. But speaking of, we talk about waterproofing. Have you ever stayed in the water too long? What happens to your fingers? They prune up. You ever seen that? Why does that happen? Do you know why that happens? Huh? It is an evolutionary advantage to help with grip, spoken by the pitcher in the back row. No, exactly. What scientists have discovered is that people that have nerve damage to their hands, no matter how long these individuals are in the water, their fingers do not prune. But that is an evolutionary adaptation to help us grip wet objects. I only discovered that just a few months ago in reading an article. You throw a spitball, is that how you know that? Okay, I'm going to watch you. So you may think that is your fingers, water, going into your fingers and causing your skin to prune up or swell up. No, it's actually your nerves and the muscles in your finger making that happen. Now, regulation of body temperature. Your skin isn't doing this on its own. Your skin, along with the blood vessels coursing through the skin, are helping your body maintain a stable homeostatic body temperature. Now, how many people are warm in here? I know this room is usually warm. Anybody warm in here? How many people are cold? Okay, different body temperatures. So we'll start with cold. More of you cold. More people were hot in the last class. I think the air came on. So if you are cold, the blood vessels in your skin are going to constrict. Less blood is going to flow towards the surface of your skin so that more of your body heat is going to be conserved in your core. One pinnacle example of that, if you are out, think about the settlers, the Donners Pass, going over the mountains in the blizzard. They got trapped and stranded. They were in the blizzard. If you're out in that environment and you don't have gloves and you don't have shoes, what's going to happen to your fingers and toes? Frostbite. You ever heard of frostbite? When you get frostbite, what color do your fingers and toes, what color do they turn? Black. You ever wonder if you're just freezing your fingers and toes, why does it turn black? The reason it turns black is because the blood vessels that provide the blood to your fingers and toes, where you would lose a lot of body heat, your blood vessels absolutely shut off. Those appendages undergo necrosis. They die. It's not that they just freeze and that's why they turn black. Because your body is going to conserve that body temperature, that core body temperature for your heart, in your brain, in your lungs. Now, when we get too hot, in addition to the next one, two down that we're going to talk about, our blood vessels dilate, they open up, and the blood courses closer to your skin, thereby providing a little bit easier way for the heat to be dissipated out of your body, kind of like the radiator of a car. That's what dissipates the heat from the engine. So you can think of your blood vessels in your skin almost like a radiator that can open up to let the heat escape or close off to keep the heat internalized. So again, skin regulates body temperature. You didn't know the skin did this much, did you? Reception. Now this is probably a no-brainer. Your skin can detect so many different signals and provide you information about your environment. We've already done one, right? What environmental interpretation did we already do when I asked you if you were hot or cold? Your skin, you sense temperature, right? What other senses can your skin detect? Pain. That's a big one. Texture, pressure. So these mechanical stresses we can detect. And pain, I don't know about you, I'm allergic to pain. I do not like pain. I avoid pain at all costs. But is pain a bad thing? It's hard for me to say that because I think it is, but no, it's not a bad thing. And I'll give you a practical example. Not only can we detect temperature, but we can detect excessive temperature. And my example in this experiment that I do probably a couple of times a year, and I don't know why it's a cookie sheet, but it's always a cookie sheet. Either I cooked a pizza on it or I cooked some biscuits or something. But if that cookie sheet is sitting there, I have to grab it. Has anybody else done that? But you grab that cookie sheet right out of the oven. Now, when you grab that hot cookie sheet, does your brain go, wow, that's hot? I can feel the flesh melting off my fingers. You know, maybe I should let this thing go. Ow. No, that's not how it works, right? You grab the cookie sheet, your hand jerks away reflexively, you realize what was going on, you utter a few expletives and look for your children and hope they didn't hear, right? Because you sense that pain. And the pain is that signal that says, idiot, stop what you're doing before you make it worse. So pay attention to pain. It's important. As are all the other senses that we have about our environment that help us survive. Now, we talked about heat and the ability to dissipate heat. Certainly in this part of the world, we understand sweat and how sweat helps cool our bodies. The evaporative nature of water, the dissipation of the heat during the evaporative process. But did you realize that sweat is also an excretory function to help remove waste? Do you know that? What waste are we removing when we sweat? I heard salt. Salt. Sweat glands are modified salt excretory glands. There's some marine organisms, birds, they have glands that exclusively express excess salt. These are modified glands that are very similar to those, which also help cool our bodies. Now, I don't think we're ever going to have so much salt that we sweat so much we dehydrate. That's not the reason we do that. But here in this part of the world, you know, August, September, when you're out and it's 105 degrees, you sweat. And you sweat a lot. Now, do we have any athletes in here? Athletes? Okay. Have you ever been in a tournament, playing so many games, playing so long, you stop sweating? Have you ever hit that point yet? That is a freaky, scary place to be. Because it's at that point you start getting weak and lightheaded, and you're like, I'm burning up and I should be sweating. Why am I not sweating? You have to get hydrated immediately. Now, don't let one of your teammates take an ice chest or a cooler of water and dump it over you and think that that's going to save your life, because it's probably going to put you in shock. The best thing to do when you are hot and you're getting to that point, other than hydrate, run some cool water over your wrists. Look at your wrists. What do you see? You see a lot of superficial vessels. That's another place where you radiate heat, and by running the cool water over your wrists, it's going to cool you down slowly, but still rapidly enough that you're going to be okay, but not too fast. So that's the whole kind of good, bad thing about the sweating and how we regulate body temperature through those processes. Now, I'm going to go ahead and warn you about the last one. Absorption and UV light. Monday, when we get toward the end of lecture on Monday, I'm going to get on my dad's soapbox. And so Monday for some of you might be the day to miss if you're going to miss a day. Because as we talk about skin and we talk about UV light, I go back to my experiences for 7 years at the Arkansas Cancer Research Center. You know where we're going? You know where I'm going here? You tanning bed people. Now, I'm going to go dad's soapbox on you, because when I was coming up, I was in the sun all the time. So it's going to be one of those dad things, do as I say, not as I do. Don't you love it when your dad does that? Well, why can't I do it? You get to do it. Shut up, I'm the dad. Right? Isn't that how it works? You hate it now, but I promise when you're a parent, you're going to catch yourself saying it. I promise you right now. Because your parents have already cursed you. I hope you have kids that act just like you. It works. I had 3 of them, and they did. UV light. UV light we associate with cancer, but UV light is essential for homeostasis of your body. Because without ultraviolet light impacting on the cells of your skin, you cannot synthesize vitamin D. Vitamin D is one of those components required for the normal development and normal growth and remodeling of your bones. And so vitamin D is essential. So you have to have some ultraviolet light. Now, don't go get a membership at a tanning salon and say, well, I need my vitamin D. Dr. Langford said it was okay. No. No, you need some. Remember, anything in excess is bad. And so we look at these things like vitamin D, and one way we know vitamin D is important is because anytime the government gets involved in something, you kind of have to stop and think, well, that must be important. Because where do we get a lot of our vitamin D these days? Well, within the sun, okay, we get it from the sun, but what product do we purchase and consume? Milk. Have you ever looked on your gallons of milk? Vitamin D fortified milk. Can you buy milk that's not vitamin D fortified? You might have to go to a farmer's market and get some of the natural straight out of the cow milk. But if you're going to go any store and get milk, it's vitamin D fortified. Because by and large, we're not out in the sun as much as the farmers back in the day. What's something else that the government deemed important that we didn't get enough of that they put in our food that we like to eat? How many of you like salt? How many of you put salt on food before you taste it? Those I don't understand y'all. I don't understand y'all at all. I got to taste it first and then put a little, What comes in your salt? Have you looked at that container of salt that you buy? Iodized salt. Do you know what iodized? They've added iodine to it. It's not that you're buying salt that already had iodine in it. No, they put it in there. Because you need iodine for your thyroid gland and the normal function of your thyroid hormones. So since we weren't getting enough in our diet any other way, they stuck it in the salt.
People like me who have high blood pressure and can't have salt, that's in literally everything you eat.
Okay, now, high blood pressure, you can't have salt. So I'm going to ask you this. My dad has high blood pressure. I got that gene too. Thanks, Dad. So I talked to my dad, how's your blood pressure? Well, it's not really getting any better. Well, Dad, didn't the doctor tell you to start using the low sodium salt? He goes, yeah, I'm using it. I'm like, well, why isn't your blood pressure getting any better? He goes, I don't know. I went home and we had a meal. So he's got the low sodium salt. He uses 10 times as much. You do that? I bet you do. Don't lie. All right. So we're on the same page with UV. You need some. You don't need too much. Because ultraviolet light impacting on our skin, and now we get into the layers of our skin, it's essential, but it can cause damage, especially to the cells that are undergoing cell division. Now, when we look at our skin, our skin cells proper, and the cells that are dividing to making new skin cells, they're going to be in the top layer of our skin called the epidermis. How do you think the people that named these two layers came up with these terms, epidermis and dermis? We've got the dermis, and then we've got this layer right above it that's epidermis. What do you think epi as a prefix means? Above. That's exactly right. It's like we got a layer and the layer above it. That's what they named them. Dermis and epidermis. Our epidermis, this is going to be our epithelial layer. And from your lab and histology, we know that there are multiple layers. We're going to label 5 layers. So how would you classify the epithelium of our skin? Multiple layers, we know that is a what? Stratified epithelium. And when we look at our skin, the surface layer of the stratified layer are fried egg, flat-looking cells. So it's a squamous, stratified squamous epithelium. Now we're going to add a word to it here in just a few minutes that's going to take into account that those top layers are also dead, keratinized. Oh, y'all got that in the lab. Oh, cool, we can skip on. But that's our epidermis. Now the dermis beneath is going to be our collagenous connective tissue layer. It's in this layer for some of our skin types. We're going to see sweat glands, hair follicles, smooth muscles, nerve endings. That's where really the business end of our skin is. Now, the layer beneath the dermis is named because the prefix hypo means what? Below, beneath. This is a third layer that some people confuse with the skin, but it's really not part of the skin. This is going to be our connective tissue that connects the skin to the rest of the body. And this is actually the layer where we're going to have some larger blood vessels, where we inject medicines, where we give vaccines. Because what do you call that needle that's used to do that? A hypodermic needle. It goes in the hypodermis, it goes below the dermis. Now, I love cool sounding words. And science is full of really cool sounding words. So your skin is also going to be that organ in that location where we have identifying characteristics for each individual. These little ridges that we have on our fingertips. And what do we call those ridges? Fingerprints. The fancy name for our fingerprints is dermatoglyphs. Sounds like hieroglyphs. Hieroglyphs, you know, the Egyptian pictorial language. Well, these pictures, these swirls, these lines denote that we're individuals. And what does derma as a prefix mean? Your skin, because if you need to go to a skin doctor, who are you going to go to? A dermatologist. So dermatoglyphs are these lines, these symbols of your skin that identify you as a unique individual. So here's our illustration. We can see the integument comprised of the epidermis and dermis. Now, subcutaneous, cutaneous also is a word that largely means skin. So subcutaneous, below the skin, is also our hypodermis. Now, what's all this yellow stuff down here in the hypodermis? Fat. You're going to see quite a bit of fat in the connective tissue of the hypodermis. Now, in some slides, depending on the individual, you can see some fat also present in the dermis, but largely that hypodermis is where it's going to collect first. Now, as we look at the line between our dermis and epidermis, we're going to see these ridges that extend up and around. These are called papillae. And when you see the papillae, that's where you can follow that sort of snaking serpentine line along to find the separation between the epidermis above and the dermis below. So, epidermis, stratified, squamous, keratinized. Where do you think we got that name keratinized? You think it means dead cells? What do you think keratinized means? It's full of keratin. Keratin being the intermediate filament that's present in these epithelial cells. And as these cells move closer and closer to the surface, they lose their nuclei, they lose their organelles, so you basically have plasma membranes filled with what? Keratin. And that's it. Keratin. Now, when we look at our skin, we're going to define it and characterize it based on its thickness or thinness. So thick skin, like we see on the palms of our hands and the soles of our feet, is going to be very distinct from the skin over the rest of our body that we call thin skin. Look, there's almost a tenfold difference in the thickness. And principally, the layer that is different in thickness is the dead cell layer on top, the stratum corneum, and we're going to get to these layers as we go through. So we'll start with thick skin first, palms and the soles of our feet. We talked about 5 layers to the epidermis. Well, the thick skin has all 5, but what it doesn't have are hairs, hair follicles, and these little smooth muscle bundles that are connected to our hair follicles called erector pili muscles. And when erector pili muscles contract, what do you see on the surface of your skin? Goosebumps. Now, sebaceous glands are these secretory glands that empty their contents into the hair follicle to try to maintain what is referred to as the suppleness of your hair. And sebaceous glands produce a waxy substance called sebum. But since we don't have hairs, we're not going to need erector pili. We don't have sebaceous glands. Those are all missing. But where it says sweat glands, I want you to put a question mark by that. And the reason I want you to put a question mark by sweat glands, these thick skin layers do have sweat glands. I think they just don't have as many as you would see in the thin skin over the rest of your body. Because have you ever been nervous and the palms of your hands get sweaty? And as we're going to look at even some of our illustrations and histological pictures of thick skin, you can see the ducts of sweat glands. So they're present, it's just not as common to see them in thick skin as you would thin. And in fact, these dots that you see right here, those are sections through the ducts of a sweat gland that is in the deeper layers of thick skin. And if you could count this up, I mean, there may be 50 plus layers of dead cells that make up this top stratum corneum layer of thick skin. And certainly here we see our papillae as it comes down, and where you see these pegs, they're often called the peg layer or the papillary layer. You can see the distinction between the darker epidermis and the lighter connective tissue dermis. And that's one way your eyes can go straight to that point to see the difference in those layers. Now, thin skin, it contains all those structures that thick skin lacked. Now, you notice we don't have anything about any sensory receptors, anything about blood vessels, because you do have those. And in some of your fingers and toes, if you've ever stepped on something sharp, you know the bottom of your foot's very sensitive. You'll also do that experiment as a parent. especially at 2 o'clock in the morning when you're trying to walk through the house to get a bottle. Yeah. But the thing about thin skin, it's missing one of our 5 layers, the stratum lucidum. The stratum lucidum is missing. And in fact, that top layer, the dead cell layer, is so thin that it practically encompasses both. But for our purposes, we're just going to say the lucidum doesn't exist. It's not present. It's not identifiable in thin skin. Now what I want you to do is I'm going to show you that previous slide again, the thick skin slide. Okay? Okay. You see the dead cell layer? And you see down here, that is the dermal boundary. See how thick that is? Okay? That dark line that you see, that's the entire epidermis of thin skin. You see the difference? That's why we talk about a tenfold difference. From that dark line down, we're looking at dermis. And from about this middle region down, as we get below the hair follicles, that's going to be our hypodermis. Look at all the fatty tissue that's present in the deeper parts of the dermis. So we can have some of that adipose tissue, right? Adipose, we're good with adipose tissue. We can have adipose tissue in the dermis, but principally we see it most often down in that lower hypodermal region. All right, so layers of our epidermis, the 5 that are present in thick skin, all of these are composed by and large of the most prominent cell type called a keratinocyte. Cyte means what? Cell. Why do you think we call this a keratinocyte? Cells filled with keratin. I mean, it's pretty straightforward. These keratinocytes are going to start forming at the very bottom layer. And we've numbered 1. That's the bottom layer, the stratum basal. And as these cells form, they're going to begin to change their shape. They're going to lose their nuclei. They're going to lose their organelles as they go from a square cell at the bottom to a flat, fried egg, dead cell at the surface. And that change in that process is called cytomorphosis. Morpho meaning form, cyto meaning cell changing its form and its shape. But that's going to be happening all through these 5 layers, from the bottom basal all the way to the top, the stratum corneum. So here we have our artist rendition on the right. We have our light microscopic picture on the left. And even though most books say thick skin doesn't have sweat glands, you can see it in the micrograph, and the artist actually put one in right there in the illustration. So they do, just not as many. So the first layer that's easiest to find, that's our dead cell layer, the one on top. And you can see how incredibly thick. So we're looking at thick skin in this illustration. We can also go to the bottom, and you can see how some of our cells look really elongated, like columnar, taller than wide. And then as we come over, some look just really kind of square, like cubes. That's cuboidal in shape. And what you have to keep in mind as you're looking at slides, you've got things that are not sitting in the perfect right orientation. They might be bent, they might be twisted, so that plane of section, you sort of have to interpret those things. The bulk of cells we see in the middle here, these are going to be the cells of our second layer, the spinosum layer. The darker region we have, these are going to be cells that contain dark waterproofing granules called the granulosum. And then we have this light layer that's trying to be illustrated, which you can't really see all that well in the micrograph. That's going to be the stratum lucidum. So, basal first. The important thing to remember about basal, this is where our keratinocytes are dividing. These cells are undergoing mitosis. And it's almost like a conveyor belt. They're formed at the bottom and they keep pushing up and up and up and up as more and more new cells are formed at the bottom. A germinal layer, like you see another name is stratum germinativum, a germinal layer is also a word that's used to describe cells that are undergoing mitosis or germinating, like you might use in a plant connotation. Now, these cells are going to stay connected to each other all the way to the surface. They're only going to lose these connections at the very surface where they can be shed. But these lateral connections between epithelial cells are called desmosomes. Did y'all talk about desmosomes in lab? Desmosomes are like Velcro. One cell has the hooks, the other cell has the loops, and they're for strength. They hold the cells together. Now, the cells along the bottom, the ones that are dividing, they also have these Velcro-like connections, but they're called hemidesmosomes, which is half of a desmosome. So down on the bottom, you basically only have the hooks that are in the cells, and those hooks attach to the proteins in the basement membrane underneath. The basement membrane for epithelial cells is just a layer of molecules that the cells sit on and anchor onto, much like a house sits on your foundation or a basement. And that's how it got that name. So we have these lateral connections to each other. We have these bottom connections to the ground underneath us. But these cells are dividing. Yet they only divide at night. Now, why would you think that would be significant enough to mention in a textbook and to mention enough in a class? These cells only divide at night. There must be some evolutionary advantage to doing that, otherwise they wouldn't be doing it. Why do your skin cells only divide at night?
When you're in REM sleep, and it's like easier to get stuff.
It's when you're asleep, what conditions are the room in typically while you're asleep? It's cool and it's dark. Your skin cells divide when there's less ultraviolet radiation that might damage what? What are we messing around with when we're dividing and replicating in the S phase? DNA. There's less ultraviolet radiation that might damage the DNA as we're undergoing cell division. Now, this is the thing I don't know. Is that a consequence of daylight, dark, or is that a consequence of wake-sleep cycle? I would love to know someone that works a graveyard shift. Do their cells only divide when they're asleep in the daytime? So is it a circadian rhythm or is it actually a light-dark cycle? I don't know the answer to that. Somebody Google that up and let us know on Monday. I'm sure somebody's done that experiment somewhere. So again, here's an illustration showing our epidermis. We've got our basal layer, single row of cells. And you can see here that's sort of highlighted in yellow. But then the next one up, the bulk of the cells we see in this area, the green number 2, that is going to be the second layer called the stratum spinosum. Those desmosomes that attach adjacent cells together, they stay intact. They're proteins in the membranes of these cells. And early histologists, as they would prepare these tissues to look at under a microscope, one of the things that they actually do is dehydrate the tissue in alcohol. It's a preservative mechanism. And as you put tissue in alcohol, you're going to dehydrate the tissue. You're removing all the water. And as they did that, the cells shrank. But as the cells shrank, they stayed attached to each other, leaving these little spines all on the surface of the cells connected to each other. And so the keratinocytes looked spiny, and that's how we got the name spinosum. And that's how they also got the cool name prickle cell, because they look like a cactus. They look kind of prickly. So this thickest of our epidermal layers, and I'm going to say thickest layers of living cells, because sometimes that top layer can be even thicker. So the thickest layer of our living epidermal cells is going to be the stratum spinosum. And sometimes near the bottom of the spinosum, where it attaches and is close to the basal, some of those cells are also undergoing mitosis. So you can have a little bit of that happening as the cells transition up into the stratum spinosum layer. Now, this is kind of a weird looking picture. It's an electron micrograph of keratinocytes in the spinosal layer. And can you sort of imagine the white space between the cells right in here? But you see how it looks like a dash line? It's not a continuous white line because it's kind of broken up by these little stripes. Those are the connections between one cell and the next. And you see how it looks kind of spiny, kind of prickly, maybe even hairy? At the light level, you can even see those little spines coming off. And again, someone said, hey, that looks like a cactus. It looks kind of prickly. So that's how those cells got that name, prickle cell. And the whole layer was called for the spiny appearance, the stratum spinosum. Now, as we get close to the top, this is the last layer of our cells that are going to have nuclei. But the thing these cells also have is they have a lot of vesicles that are filled with material, and this material is very rich in lipids. And these vesicles, as they coalesce together, are going to help create a waterproof barrier. Now, why would you maybe suspect that lipid materials might be used to waterproof a layer? What's another term that we could use to describe lipids and hydrocarbons, non-polar molecules? We could say they are what? Hydrophobic. Hydrophobic. And so these hydrophobic lipids in these vesicles are going to help begin to create a waterproof barrier. Principally so water can't get in, but also it prevents the desiccation and water leaving. So stratum granulosum, last layer with nuclei, last layer of living cells, but it's also important in the waterproofing of our epidermis. So here we can see the second spinosal layer, but look how much thicker the top layer is. That's why the spinosum is the thickest of our living layers. But can you also see this dark line running right along? To me, the granulosum is almost the most easy to identify. When I'm teaching my senior pre-med histology students and we look at these pictures, my eye immediately goes to the granulosal layer. And that helps me distinguish the layers on top from the layers on bottom. Speaking of on top, when you identify the granulosum, you know just above it, even though you may not be able to see it all that well, is the stratum lucidum. There are no nuclei in the cells, in the keratinocytes in the lucidum. In fact, there are no organelles. The only thing left in this process of cytomorphosis are parallel rows of keratin filaments. And these parallel rows of keratin filaments allow more light to easily pass through. And that's why it looks like a lighter zone or a lighter band as you're looking at histological images and sections of skin tissue. So as the cells are pushing up, the nuclei are simply degraded. They're simply degraded. They're lost. It's not like they get spit out, like we'll see red blood cells and red blood cell development next semester. Red blood cells in their development, they eject the nucleus. These just degrade it. They just degrade it to get it out of the way. So no nuclei, no organelles, filled with keratin. And remember, keratin is a type of intermediate filament. Now, intermediate filaments, that's one of three types of what molecules present in a cell? What category would you put that in? Somebody said it. Cytoskeleton. So keratin intermediate filaments is a cytoskeletal component. Along with microfilaments, and what's the biggest one? Microtubules. Those are your three cytoskeletal elements. Now this is a different stain. This is almost an exclusion stain because we can see the nuclei of our basal layer, of our spinosum layer. We can't really see the granules, but you can see the nuclei, and then all of a sudden the nuclei are gone. But we can see the distinction between this lighter region and then this much more dense region. This is the stratum lucidum, just above that last layer of nucleated cells in our epidermis. That's also going to assist in our waterproofing of the skin. Now the corneum, this is the top. This is the last layer. These are the plates of dead cells. And this is really going to assist in making a pathogen boundary. Because when you think of pathogens like virus, they need living cells to replicate. Well, that's not going to help them to infect these cells because they're dead. Not only that, but these cells are continually losing their connection to each other. Those desmosomes break down. And when they're not connected to each other, it's a lot easier for them to be shed. And since we call these flattened cells squams, the shedding of your skin is called desquamation. Now, on Monday, when we talk about burns and sometime having to remove your skin that's been burned so severely, that's called debridement. That's very different than losing the surface layer of your cells, which is called desquamation. But again, we've seen a number of pictures already today, especially in our thick skin. But here you can clearly see all these rows and rows and layers of our thick skin.